I want to thank the workshop organizers for this opportunity to present our research on antimicrobial resistance in the rivers and streams of the United States. Antimicrobial resistance, or AR, is a serious global One Health problem due to the overuse of antimicrobials in human, animal, and agricultural sectors. AR is naturally occurring in the environment and it is important to establish background levels to distinguish it from AR due to point and non-point discharges as described in the graphic on this slide. It is unclear whether increased abundance of ARGs in the environment is simply a result of fecal contamination from human and animal agriculture, or if antibiotics in the environment are selecting for resistant organisms, or if ARGs are transferred between bacteria and the environment. There is a research need to better understand the ecology and environmental processes influencing the spread of AR in the environment. Our approach to address increased AR in the environment is to leverage the National Rivers and Stream Assessment, or NRSA survey, and determine the concentrations of ARGs in these waters. NRSA provides information on the ecological condition of the nation's rivers and streams and the key stressors that affect them at national and ecoregional scales. The survey is completed every 5 years to monitor changes in condition. The 2013-2014 cycle of the NRSA included 1,853 sites to represent 1.2 million kilometers, which is possible because the survey is a stratified random design to produce balanced sampling across the country. Each selected sample is assigned a weight to represent a given number of river or stream kilometers. This slide shows the boundaries for the 9 ecoregions in this study. These ecoregions have very different ecological features. For example, 69% of the temperate plains is used for crop production and animal agriculture, whereas 54% of the Western Mountains is forest and 37% of it is grasslands and shrubs. The sample sites are colored to indicate least disturbed, blue, intermediate disturbed, yellow, and most disturbed sites, red, in the 9 ecoregions. The approach for sample collection and PCR is described on this slide. 50 ml water samples were collected at base flow conditions from weightable and non-weightable waters. Frozen filters were sent to Cincinnati labs for a US EPA Method 1609 PCR analysis. We used a portion of the DNA for digital PCR to determine the concentrations of target genes adjusted by the survey weights to provide weighted means per milliliter. The selected genes in this study are shown on this slide. We selected the integron integrase gene because it is regulated by the SOS response system, is involved in mobility of ARGs, and has been used as a proxy indicator of anthropogenic pollution. The SOL1 gene was selected because it is commonly used in environmental studies, and sulfonamides are classified as highly important by the World Health Organization. TETW was selected because tetracyclines are also highly important and one of the most widely used drugs in food production. We selected BLATEM because beta-lactams are widely used to treat human infections. Approximately 2/3 of antibiotics administered to humans are beta-lactams. The other genes were selected because they represent drugs of last resort. We must be mindful that there are several other classes of ARGs to choose from, as the graphic indicates, but because of the number of sites in NRSA and budget constraints, we selected this list of genes to complete the study. This slide shows the results of the ARG survey. It also contains the results for 2 fecal indicator bacteria and total bacteria. This figure is a heat map of the weighted means for the 7 gene targets and shows the rankings by ecoregion. The light to dark blue color gradient indicates the weighted means per milliliter. In general, the temperate and coastal plains ranked near the top for most of the genes, whereas xeric, northern plains, and northern Appalachians tended to be near the bottom of the rankings. The Western Mountains came in dead last for all genes. Not surprisingly, the rankings were similar for anti-1 and SOL1. This is likely due to the genetic linkage of the 2 genes in class 1 integrons. What is causing high gene concentrations in the plains? Let's describe the type of land use and review the nutrient and fecal pollution levels for temperate plains and compare it to Western Mountains, using condition indicator data from the interactive NRSA dashboard that is publicly available using the link on this slide. The two screenshots from the dashboard are total nitrogen on the left and on the right Enterococcus water quality criterion for protecting human health. The point estimates on the horizontal bar graphs indicate the percentages of river and stream miles of poor quality for each ecoregion. The vertical gray area passing through the bar graphs is the national average. The temperate plains are primarily agricultural land for crop, hog, and cattle production. The EPA found that more than half of temperate plains, river, and stream miles were of poor quality for total nitrogen and total phosphorus. For fecal pollution, 38.5% of the miles were above the health criterion. The Western Mountains are mostly comprised of forests, grasslands, and shrubs. The EPA found that only 20.5% of Western Mountains, river, and stream miles were poor quality for total nitrogen, which is a third of what was reported for temperate plains. However, total phosphorus was similar to temperate plains. And finally, only 15% of Western Mountains miles were above the health criterion for fecal pollution, which is less than half of the temperate plains. These ecoregional differences in nutrient and fecal pollution suggest rivers and streams in poor condition will contain higher ARG concentrations than those in good condition. Point and non-point sources of nutrient and fecal pollution are likely to be the sources for elevated levels of ARGs above what is expected for background, or what we refer to as least disturbed conditions. Because natural river and stream quantities of ARGs and environmental bacteria may vary across the country, we compare their quantities for least and most disturbed conditions within each ecoregion. The least disturbed sites were selected because they represent the best range of conditions that can be achieved for similar rivers and streams within a particular ecoregion. For more information on this topic, please consult the NRSA EPA report using the link at the bottom of this slide. The two tables show total phosphorus and total nitrogen, as well as other water and habitat parameters for least disturbed and most disturbed conditions. This slide shows the results of the comparisons. Plots for all 7 genes are shown to the left. The heat map summarizes the magnitude of the differences between least and most disturbed sites. The heat map scale is log 2. The Anti1 and Sol1 genes support our hypothesis that rivers and streams in poor condition have high gene concentrations. Most of the ecoregions support this conclusion except for northern and temperate plains. National differences were observed for TET-W and BLA10, and only a few ecoregions were different for these genes. Interestingly, there was no national difference for the fecal indicator Enterococcus, and only a couple of ecoregions were statistically different. Both national and ecoregional differences, 5 out of 9, were observed for E. coli. National and ecoregional differences were observed for 16S ribosomal RNA gene. This result underscores the importance of understanding the role nutrient pollution plays on the growth of aquatic bacteria, and the need to account for bacterial growth when assessing ARGs. The conclusions for this presentation are the following. ARGs showed significant geospatial patterns at national scale. Good quality rivers and streams had lower ARG concentrations than poor quality ones. These data suggest Anti1 can be used as an operational ecological condition indicator. Other types of baseline analysis gave similar results to river and stream condition analysis described in this talk. Urbanization and poor watershed integrity were significantly associated with high concentrations of Anti1 and Sol1. Poor watershed integrity, but not urbanization, was associated with high concentrations of TET-W. Urbanization and poor watershed integrity were not associated with BLA10. Next steps. Statistical modeling research is ongoing. The Interagency NARNS program is expanding AR research to include the environment through a collaboration with the EPA. We are expanding NRSA analysis using the Fluidine system to screen for new ARG targets. We are including ARG measurements in the next cycle of NRSA, and we are exploring metagenomic resistance sequencing. So to continue with next steps, I want to show some preliminary data using the Fluidine system. We have a customized panel of gene targets that cover resistance genes to drug classes such as aminoglycosides, beta-lactams, macrolides, tetracyclines, and other drug classes. This slide shows that impaired sites, or what we've been calling most disturbed sites, have higher proportions of positive PCRs than do least disturbed sites, here labeled reference. Heat maps of the replicate runs are shown in the upper right. To continue with next steps, here we show preliminary metagenomic data for an ecoregion of the NRSA. This slide shows a network graph comparing least and most disturbed sites, labeled R and T. ARGs unique to each condition class are shown to the left and right of each condition class node. The graphs on the right show the abundance of sequence reads by drug class broken out by year. Resistance genes for aminoglycosides, beta-lactams, macrolides, and tetracyclines were unique to the most disturbed sites in the network graph, and their read abundances were higher in most disturbed sites. This research is a collaborative effort involving the NRSA Steering Committee, as well as state, tribal, territory, and interstate partners. I would like to acknowledge the ORD team, the Office of Water, and the many individuals that collected the samples and supported this survey. Thanks for your time.